Okay, so today we're gonna only do one chapter, which is Form 4, Chapter 2. We will be doing only one chapter because we only have 2 hours lah. For those who don't know who am I, my name is Wai Ling. I'm the one on your cover page. Looks the same, right? Okay, I'm the person on the cover page. So the reason why I choose chapter 2 instead of chapter 1, because chapter 1, they will only ask in your first term exam. So as you go to midterm, as you go to, not midterm, your final exam, or maybe your Form 5 or even your trials, you will not see Form 4 chapter 1 anymore. So it's only important for the first term for your Form 4 exam. But for Form 4 Chapter 2, you will see until SPM. So Chapter 2 is actually very important. If you don't know Chapter 2, actually there will be a lot of chapter that you don't understand, okay? So in Chapter 2, what do we have to learn? We have to learn about plant cell, animal cell. We have to learn the things inside plant cell, animal cell. Okay, first, how do we differentiate which one is animal cell or plant cell? We have to look at the outer membrane. Usually for plant cell, there is a thing called cell wall. So there's cell wall, but animal cell has no cell wall. Why? Because plant cell wall is to maintain a fixed shape and they are tough and rigid. So usually they don't collapse so easily. Animal cell, they have no cell wall. They are irregular in shape. So the second difference between plant cell and animal cell. Anybody know what is the difference between animal cell and plant cell? Correct. Animal cell has centrioles. This is actually a centrioles, okay? They have centriole, but plant cell has no centrioles. It's to form spindle fiber during cell division. Okay, 3, they have chloroplasts. Very good. Plant cell has a chloroplast, so that's why they have to be green in color. All plant cell has to be green in color when they have chloroplast because they have chlorophyll. Animal cell has no chloroplast. They are not green in color. And vacuole. Can you write no vacuole and be vacuole? Actually, animal cell has a large vacuole. Plant cell has a vacuole, but they are small. So we don't say no vacuole and large vacuole. We will say large vacuole and small vacuole because animal cell has vacuole as well. So we have 3 differences actually. 3, 4. Okay, we have cell wall, we have chloroplast, we have centrioles, and also vacuole. All right, large central vacuole. Some teacher, okay, some school teacher, I'm not saying that all school teachers, some school teacher, they don't want you to write fixed shape and no fixed shape. Some school teacher says that you have to write plant cell has a fixed shape, whereas animal cell has irregular in shape. Irregular, not no fixed shape. If your teacher are so specific over this answer, then you write irregular in shape. Large central vacuole. You can write large vacuole is enough already actually for differences. Okay, so this is the differences. Now we have to learn what is inside the animal cell and plant cell. We have organelle and non-organelle. Okay, what is organelle? What is non-organelle? Okay, that stores carbohydrate. Okay, the method of storing carbohydrate is different as well. So for animal cell, they will store as a glycogen. For plant cell, they will store as a starch. Usually, if you want to write differences, you can choose any 3 that you know. Organelle means what? Organelle, it means cellular component. Cellular component. Okay, cellular component are the organ in cell. Organ in cell. We call this as cellular component. Organelle in, okay, it means organ in cell. For non-organelle, they are not in the cell. Non-organelle is over here, right? Yeah, not in cell. What is not in cell here? We only have 3 examples. We have plasma membrane, cytoplasm, and cell wall. We start with plasma membrane first. Plasma membrane is also known as a cell membrane, but in Form 4, we cannot write cell membrane. We can only write plasma membrane. Okay, remember, only write plasma membrane, not cell membrane. Okay, is cell membrane inside the cell? Is cell membrane inside the cell? No, right? They are outside, right? So that's why they are not organelle. Second, cytoplasm is the space. The space is a space. They are not anything inside the cell. So cytoplasm is also non-organelle. Cell wall is outside, the most outer layer of plant cell. They are not inside the cell also. We can imagine this like a building. Okay, a building is like cell wall. Okay, a building is a cell wall. And then we have a classroom. The classroom is a plasma membrane. The spaces in the classroom is cytoplasm. Okay, imagine your house. Your house is a cell wall. Your room is a plasma membrane right now. The spaces in your room is a cytoplasm. So what is in your room? You have table, chairs, your bed, your water bottle, your laptop. Those are the things inside your room, right? Those are called organelle. Plasma membrane has characteristic and function. When you see the label, which is this symbol over here, right? This one is the characteristic. I put 2 different symbols, right? This is the characteristic. This is the function. The characteristic of plasma membrane, they are selectively permeable and they are partially permeable. I will not suggest you to write semi-permeable because Your teacher, some teacher, I cannot say all guys, different teacher, they have different pattern, okay? So some teacher, they don't want you to write semi-permeable. They only want you to write selectively permeable. We learned this in chapter 4 and chapter 3, and then partially permeable in chapter 3. So actually the best answer in SPM, we only write selectively permeable. This is the best answer to write. Unless your teacher says, okay, you can write semi-permeable. If it's not, selectively permeable is your only choice, okay? Selectively permeable. They will only allow certain substances to pass through. Missing word here, of certain substances. Certain is the keyword. Certain means what? Only small thing can goes in, the large one cannot goes in. The small ones such as water, gases, they are able to diffuse in and out. But for glucose, amino acid, they cannot, because they are selectively permeable. Okay, remember, no cell membrane is called plasma membrane. Then we have cytoplasm, is a jelly-like structure. This cytoplasm is a place where biochemical reaction occurs. Bio means living thing. Chemical reaction happens in the living thing. We call this as biochemical reaction. Any chemical reaction that happens in human body is known as biochemical reaction. Example, in Form 4, Chapter 4, we learn about hydrolysis. How to digest food is a biochemical reaction. To digest food from starch to maltose, maltose to glucose is a biochemical reaction, okay? Cell wall, they are made of cellulose. If you remember, cellulose is a sugar that is not sweet. This is a sugar that is not sweet, okay? A sugar that is not sweet, we call this as cellulose. They are under polysaccharide. Form 4, Chapter 4, carbohydrate, there's a thing called polysaccharide. All of the polysaccharide, they are not sweet, not soluble, and non-crystalline. So cellulose, they are made of a sugar. When you break down cellulose, they will form into glucose. Okay, the characteristic of it, besides they are made of cellulose, they are rigid and tough. Okay, if you want to write characteristic, right over here, rigid and tough, or tough and rigid, both also okay. This is another characteristic. You can say permeable to all fluid also can. Rigid and tough is another characteristic. If you want to write rigid and tough, your explanation is they are able to provide mechanical support to plant, means to provide support to the plant and to maintain the shape of a plant. So whenever you want to explain your answer, how does a cell wall maintain the shape of a plant cell? You have to write because they are rigid and tough. So this is your statement. This will be your explanation. This is for cell wall. Okay, so this is plasma membrane, cytoplasm, and cell wall. We have learned this actually in Form 1 or Form 2 before. Right now, we're going to learn something that we started to learn in Form 4. Okay, first, I know you know what is a nucleus. Nucleus is a brain of a cell. Human, our brain is the one that controls us. If a cell without nucleus, they will die. If human without nucleus, we will also die. So nucleus is very important to all cell, no matter whether it's animal cell or plant cell, okay? They are very important. Besides red blood cell, they have no nucleus, okay? The rest of the cell, they have nucleus, yeah? They are made of double membrane. What is double membrane here? They have two layers. Outer one layer, inner another layer. So they have two layers. The function of nucleus is to control all cell activities. It contains of DNA. DNA stands for deoxyribonucleic acid. Can you write DNA in exam? If for first term, Form 4 first term exam, some of your teacher want you to write deoxyribonucleic acid, but in SPM, okay, means after your first term, after this exam onwards, you can write DNA. So how do you know whether you can write DNA, you can write short form or not? As long as the short form can be found in your book, it means it's a short form that you can write, means your textbook lah, okay? So if your teacher insists you that you have to write deoxyribonucleic acid, then you write for her. If it's not, just write DNA. So do know whether to write or not to write. Go and ask your teacher before your exam. Usually DNA is an acceptable answer. You can write DNA. You can say contains of DNA also can be no problem one. They can determine the characteristic of cell. Example, the reason why you have black color hair because your parents have black color hair. The reason why you have no blue eyes because your parents don't have blue eye. The reason why you have this height because your parents are almost the same height. The reason why you have double eyelid or single eyelid, everything comes from the parents because your parents give you DNA. DNA is the one that contains of your characteristic. You will somehow has your parents' feature because your parents is the one that give you genetic information, right? You will not look like random people on the street, right, guys? So because the DNA come from our parents, this is DNA. Next, mitochondria. Mitochondrion and mitochondria is singular plural, so it doesn't matter. The mitochondria is a powerhouse of the cell. They are powerhouse of the cell. They are able to generate energy. Mitochondria, they also have double membrane. How do we see double membrane here? This is the first membrane. This is the second membrane. Inside the second membrane, they are folded. It looks like a cloud. Okay, C-L-O-U. It looks like a cloud, right? So this is the folded membrane. Anything that is folded in biology has the same explanation, is to increase surface area. Anything that is folded in biology is to increase surface area. Your mitochondria is to generate energy in the form of ATP. Can you write ATP instead of adenosine triphosphate? Again, the answer is yes. In SPM, you can write ATP. Okay, in your first term, if your teacher say yes, you can also write ATP. No problem to write in short form. Okay, it's got in the form of ATP. ATP stands for adenosine triphosphate. It's a form of energy. It means we have electrical energy, we have chemical energy, right? So ATP can be produced in human cell or maybe in plant cell because we cannot generate electrical energy. Human, we don't generate electrical energy. If you can generate electrical energy, you're not human, guys. You're Pikachu. Okay, so we cannot generate energy in the form of electrical formula. We can generate in the form of ATP. How do we produce energy as a human? We will go through a process called cellular respiration. What is a cellular respiration? Is where they will break down glucose into energy. Okay, this process here is called cellular respiration. With the presence of oxygen, it's called cellular respiration. Your parents will always tell you, if you don't eat rice, you have no energy. Have you guys heard of this? If you eat rice, you have energy. If you don't eat rice, you have no energy. Okay, now where does this saying come from? Because your rice is glucose. Glucose will form into energy, not just rice, guys. All form of carbohydrate, which is your noodles, your bread, whatever, like, as long as that carbohydrate, they give you the same thing. We have to go through cellular respiration. Only we have enough of energy. Without energy, literally, you cannot move. You cannot grow taller also. So this is mitochondria. Okay, mitochondria generate energy in the form of ATP. Next, we have chloroplasts. Chloroplasts, they are also double membrane. Okay, it looks like I would say, twin tower kind of thing. They're like a tower. It can only be found in plant cell. Chloroplast has to be green color. Why? Because chloroplasts, they have chlorophyll. Chlorophyll, they are green in color. They are able to trap sunlight. You can write trap sunlight. You can also write absorb sunlight. They will convert into chemical energy. Chemical means what? Food. They will convert your light into chemical, means from the light they will break down and then they'll form into food, which is glucose. During photosynthesis, some of your exam, your teacher will create their own diagram, means they will draw by themselves. How do they draw by themselves? Usually for mitochondria, they will draw something like that. Okay, you can also draw it down, okay? I'll draw it over here. So it's like a oval shape, okay? And then it looks like a cloud inside. Like that, okay? This is a mitochondria, double membrane. For chloroplast, also same oval shape. Okay, and then we have another one because it's double membrane, right? Okay, oval shape. 2-layer. Inside looks like a twin tower, like Baymax eye. If you guys watched Baymax before, like Baymax eyes, this is how it looks like. In diagram, this is how it looks like. So this is chloroplast. So you have to recognize the diagram, yeah, whether they looks like a Baymax eyes or they have a cloud. This is how we differentiate mitochondria and chloroplast. If they ask you what is the similarity, guys, similarity has only one answer, which is double membrane. You cannot write they form energy. It's different, guys. You have to write double membrane. Double membrane. There's only one answer to this, which is double membrane only. Okay, both have double membrane. Okay, next we have vacuole. Vacuole is a fluid-filled sac. What is inside the vacuole? Inside the vacuole, we have cell sac. Cell sac means what? Cell sac means that they have nutrients, water, waste substances, glucose, amino acid gases, anything. It can be found in the cell sac. The purpose of vacuole is to maintain turgidity. Where does the word turgidity come from? Turgidity comes from a pressure. It's called turgo pressure. The pressure is to provide support to who? To the non-woody plant. They uses water as a support to non-woody plant. Okay, so without water, non-woody plant unable to maintain turgidity, unable to have the turgidity. No water means not turgid. Not turgid means no support for non-woody plant. For woody plant, they have woody tissue, so they don't have to use water. So remember, the word turgid It means water goes into the cell. The vacuole becomes bigger, they have support. We call this as turgid. Remember, turgid means only water provides support. Rigid is for cell wall, turgid is for vacuole. It's different, huh? Turgid is for vacuole, rigid is for cell wall. Both also provide support, but different form of support, okay? So this is vacuole. Okay, next, lysosome. Lysosome is like your stomach. Okay, what is the function of a stomach? It's to digest food. How do we digest food? Is by using enzyme. So lysosome will secrete an enzyme called hydrolytic enzyme or lysozyme. You can write either one, guys. You can write hydrolytic enzyme. You can also write lysozyme. But if it's me, I would choose to write lysozyme because it's easier. Lysozyme, secreted by the lysosome. It's like stomach. Is to break down food. So they will break down complex substances. They will eliminate damaged organelle. Eliminate means they want to remove. It's called eliminate. Example, if the chloroplast is not working already, there's no point for you to have another chloroplast in your stomach, right? So the lysosome, they will secrete lysozyme. They will break down the chloroplast. Example, if the mitochondria is not working already, lysosome secrete lysozyme. They will break down the mitochondria. So it's a place, it's a destroyer. I cannot say destroyer. It's a place where they remove things. It's like a recycle bin. Okay, maybe a better word is like a recycle bin. It's to remove things that you don't want, is to break down unwanted substances. This is lysosome. Okay, lysosome. Ribosome has no diagram. Why? Because ribosome is a dot. The dot can be found on the rough ER. Okay, on the rough ER, can you see that there are dots? Don't know if you can see, but there are dots over here. This is ribosome. So ribosome is the dot. The function of ribosome is to synthesize protein. What does synthesize mean? Synthesize means produce. Okay, the word synthesize means produce. Is to produce protein. As they produce protein, they attach onto the rough ER. The reason why they are rough is because they have ribosome. It's called a rough ER. They have a lot of ribosome on top. Is to transport protein. Transport, synthesize protein. Okay, transport protein. Where do they transport protein to? From the rough ER, they will transport protein to the Golgi Apparatus. I will come back to this later, guys, okay? We'll go to Golgi Apparatus first. So, ribosome attached onto the rough ER. Rough ER transport protein to the Golgi Apparatus. This Golgi Apparatus is like a factory. Okay, like a legit factory. Have you guys been to any factory before? Like a bread factory, Maggie factory, chocolate factory, okay, factory. All factory works almost the same thing. Okay, let's say right now I want to make it into bread, okay. If you want to make it into chocolate, you have to use the cocoa nut, right? And then you break and then you make it into chocolate, right? You have white chocolate, whatever chocolate, okay. But if you want to make it into bread, you have no dough at the first place. You have to use what? You have to put flour inside. You're going to put water inside. You're going to put yeast inside. As you mash, put everything inside already, right? They form into a dough. This dough right now got no flavor. It's just a normal flavor. If I want to make it into a chocolate bread, how should I do it? I use the chocolate, okay? I put it inside. I modify the dough into chocolate flavor, right? And then the bread right now is chocolate flavor bread. After I process and modify it already, right, I have to pack them. I cannot sell the bread to without packaging. So I have to pack them. After I pack them, from the factory, I will send it to maybe 99, 7-Eleven, KK So I will send it out. So it's like a factory. Golgi Apparatus is like a factory. They will process, modify, pack or sort, and transport substances. What do they transport? Is actually protein. In short, we can remember as PMPST. In your textbook, we only write package. We don't have to write sort. So if your teacher follow textbook, you can choose to write process, modify, pack, and transport, PMPT. The sequence has to be correct, yeah? So it's process come first. Pro, okay? Pro. If your teacher want everything, then you're going to write everything, okay? So process, and then you can only pack. You cannot pack first because you have nothing to pack at the first place. You need to process first. And then you modify, you pack, sort, and transport. Where do they transport to? They transport out of the cell or within the cell. So if you put it into a sequence, this ribosome, rough ER, and Golgi Apparatus, they always ask relationship. Because they're always together, right? They have a relationship. All of these three things here, they are related to protein. Ribosome synthesize protein. Rough ER transport protein. Golgi Apparatus modified protein. Everything here related to protein. And if I put it into a sequence, okay, we have to know that whenever you transport things, we need a transporter. From the rough ER to the Golgi Apparatus, we have to use a transporter. It's like from your house to your school. Either you walk to your school, is either you take a bus to your school, is either you take LRT to school, right? So this one also need a transporter. The transporter right now is called transport vesicle. Transport vesicle contains of only protein. Transport vesicle only has protein inside. Why there's only protein? Because they transport protein. Of course, there's only protein. For Golgi apparatus, they will be transported out of the cell or within the cell, right? Using secretory vesicle. Secretory vesicle, they have hormone or enzyme. Either one, okay? Because it has been modified. So they have hormone or enzyme inside. In sequence, we start with ribosome first. Okay, ribosome, they attach on RAF-ER. Okay, they attach on the RAF-ER, attach on RAF endoplasmic reticulum. Exam cannot write RER, yeah? And then they will transport protein. Transport protein to Golgi apparatus. The G has to be capital letter. When they want to transport, they have to use a vesicle called transport vesicle. Golgi apparatus, they will pack PMPST out of cell or within cell. Either one, okay, out of cell or within cell. Okay, I'm gonna write the short form on top. PMPST, okay? PMPST by using a vesicle. The vesicle is called secretory vesicle. It's called secretory vesicle. So whenever they ask relationship, always remember, just write down the function. What is the function of ribosome, RAF-ER, Golgi apparatus? Okay, so this is the sequence of it. Okay, now done with this, we're going to go to the next one, which is smooth ER. Smooth and rough. The difference here is what? Smooth, right? Okay, smooth means what? There's nothing on the surface. It's smooth. Unlike rough ER got dot, this one got nothing. It's smooth ER. They will synthesize lipid. Can you guys tell me again what does the word synthesize mean? Correct, produce. They will produce lipid means fats. They will synthesize lipid. Lipid means fats, yeah? Fats, okay? And they will carry out detoxification. Detoxification means they want to remove the toxic from your body. It's called detoxification. People who take drugs, people who take a lot of alcohol, people who take a lot of medicine, their liver usually not very good because smooth ER can be found abundantly in the liver. Like it, let me write over here, yeah? You can also write together, yeah? Found abundantly. Abundantly means a lot. It can be found abundantly in liver cell. Your liver is to carry out detoxification. I don't know if you guys heard of people who say, oh, people who drink a lot of alcohol, their liver definitely is not good. People who take a lot of medicine, take a lot of drugs, they are also not good liver, right? Because liver is to carry out detoxification of drugs. This is smooth ER, can be found abundantly in your liver cell. Okay, so smooth, rough ER, smooth ER, and Golgi apparatus. In diagram, they look very similar. How do we differentiate these three things? Let me draw for you to see. Usually rough ER, they are connected together. And this is also connected together. They have a very similar diagram, but one is with dot. Okay, smooth ER is without the dot. Smooth and rough, obviously, this is how we differentiate, right? For Golgi apparatus, literally, it looks like that. It's like a Wi-Fi symbol. Like Wi-Fi, right? They are not connected together. Just Wi-Fi symbol, not connected together. The other thing that we can know is rough and smooth ER, these two, rough and smooth ER, can be found outside of the nucleus. They are found outside of nucleus, outside of nucleus. If you look back to the diagram on top, can you see your nucleus outside is your rough and smooth ER. Your Golgi apparatus is not sticking to the nucleus. Same goes to this. Outside of it, we have rough and smooth ER. So whenever you see outside of the nucleus, is either rough or is either smooth. It cannot be Golgi apparatus. So this is how we differentiate smooth ER and Golgi apparatus. Usually these two is the most confusing one because both looks very similar. So that's why this is how we differentiate. Okay. Last, we have centrioles. Centrioles can be found only in animal cell. The function is to form spindle fiber. So it's the line is to pull chromosome apart. When they cannot pull chromosome apart, your number of chromosome is abnormal. So that's why there are people who have Down syndrome. Down syndrome maybe because the centriole cannot form spindle fiber. Then they have abnormal number of chromosome. Centrioles, it can be 3D in your question, or it can be 2D, which is only this part. The 2D, only the dot like that, a circle like that, okay? This is also a centrioles. So it's not every time it's 3D in your question, it can be 2D also, okay? So this is centrioles. So now that we have learned all of the cellular components, okay, are you guys okay with this? Anybody has question to ask in this page? Or is it too fast or too slow for you? Okay, normal speed. Yes, to you it's normal speed because you get used to it with my speed already. Okay, now the ribosome on the surface of rough ER. Okay, yeah, ribosome is on the surface of rough ER. Sometimes you can also find ribosome at the cytoplasm also actually. So it's either on the rough ER or it's either on the cytoplasm because it can be a free ribosome in the cytoplasm. How to differentiate centrioles and lysosome when it's circle? Usually dotted circle is centrioles. Lysosome is a solid circle. How do we draw? We don't have to draw centrioles. They don't ask you to draw centrioles, yeah. I'm a person who usually would get sick. That's why your liver is not good. And not really lah, guys. Not really, okay? So you have a lot of medicine. So you always get sick. You eat, or you, because you eat a lot of medicine, that's why you think your liver cell is not good. As long as you take the medicine according what the doctor give it to you, then no problem already. Exam cannot write RER. You have to write rough endoplasmic reticulum. Which organelle do we usually have to draw? I would say drawing of organelle is not a common asked question. They don't really ask you to draw organelle. Okay, out of, I would say 20 exam paper that I've seen, only one exam paper they only ask you to draw. So the thing right now that we have to know is we have to know how to recognize the diagram. Means recognize is one, don't have to draw. Number two is to know the spelling of the name. Number three, the characteristic of the name. Number four, the function of the diagram. There's only four things that you know. Recognize, spelling of the name, characteristic, and function. There are only four things you have to know. Drawing is never, drawing is not usually a question that you will see. But later we will draw a diagram. We will draw mitochondria later, okay? Okay, now, done. Okay, now next we're going to go through one more part and then we're going to try some question, yeah? Since we have learned the function already, right? This is certain of a density of certain organelle. The density here in this subtopic is to refers to high density. Okay, to refers to high density means a lot of, okay, high density. Yeah. First, we talk about mitochondria. Can you guys tell me what is the function of mitochondria? What is the function of mitochondria? Correct, they are powerhouse. Correct, generate energy. Very good. Generate energy in the form of ATP. They are powerhouse. Mitochondrion and mitochondria is the same, guys. It's up to you whether you want to write singular, plural, doesn't matter. Muscle cell, sperm cell can be found in animal cell. This one can be found in animal cell. Animal means human. Human is animal cell, yeah. This is also animal cell. Okay, and then for meristematic cell, this is in plant cell. This is one of the things that we have to take note, guys. Okay, we have to take note. Animal cell and plant cell, we have to look at the question. Sometimes they ask you to write plant cell. Sometimes they ask you to write animal cell. Muscle cell has only two actions, which is to contract and to relax. Muscle cell can only contract and relax. Okay, can only contract and relax. So they need more energy to have muscle movement. Whenever you are doing any action, when you're writing, when you're walking, when you're dancing, when you're sitting down, lying down, everything is a muscle contraction. You must have contract and relax. You cannot contract two times, relax one time. Because the moment you only contract but you never relax, you get muscle cramp. So it's contract and relax to create a movement. Sperm cell can only be found in guy body, obviously, because sperm is the one that fertilizes the ovum, right? So they need more energy to swim and penetrate. Penetrate means they will go into the ovum for fertilization process. When a guy mitochondria is not present, the sperm cannot swim, means that it's very difficult for them to have baby. Not cannot lah, but it's difficult lah. When a sperm without tail also cannot have baby because they cannot swim. Cannot have baby doesn't mean that they have no baby, guys. Cannot have baby means that they cannot have baby naturally. Maybe they can do IVF. Test tube baby, they are able to do it. Test tube baby, you need the nucleus and nucleus, right? Then they can still have their own baby. But if without mitochondria and without tail, it's not possible for the sperm to move at all. Sometimes in some sperm is deformed sperm because of mutation. Meristematic cell. Meristem means what? Meristem or meristematic is the same thing, yeah? Meristematic here refers to young plant cell. It has to be young. Okay. If no ovum have to be baby, no ovum has to be outside. Okay. Ovum have to be in the female body. They swim to the ovum. Okay. Now young plant cell here, every young thing they have to grow taller. Okay. Every young thing they have to grow taller. It's like human. When human last time you are only maybe 60 cm. Right now you're 160 cm. You grow up to 100 cm, right? How do you grow? It's because of mitosis. It's because of cell division. 1 to 2, 2 to 4, 4 to 8. Okay, they keep on undergoing mitosis, then it will only form into many, many cells, right? Same goes to plant cell. They need to have more energy to produce new cell during cell division. Cell division means 1 to 2, 2 to 4 is called cell division. So if the question wants you to write which animal cell has a lot of mitochondria, you have to write sperm cell or muscle cell. Which plant cell has a lot of mitochondria? You have to write meristematic cell. It's just a careless mistake that people usually make. It's not like you don't know, just that it's a careless mistake lah. Okay, next, chloroplast. Can you guys tell me what is the function of chloroplast? Chloroplast contains of what? The green color thing is known as? Very good. It's known as chlorophyll. It's used to absorb sunlight to carry out photosynthesis. Very good. It absorbs sunlight to carry out photosynthesis. Yeah. Which cell has a lot of chloroplast? Palisade mesophyll cell has the most, okay, they have the most chloroplast. But in Form 4, they also write palisade mesophyll cell, spongy mesophyll cell, gut cell. All of these three has a lot of chloroplast. But actually in Form 5, you will learn that palisade has more, spongy has lesser. If you want to write which cell has it, then you can write palisade, spongy, and gut cell. Yeah, this one has the most. Okay, most. This one has the least, lesser. This one also lesser. Okay, so this tree has chloroplast. They will trap maximum sunlight for higher rate of photosynthesis. Ribosome, RER, Golgi Apparatus. All of these three has the same characteristic, which is protein. Ribosome is to do what, guys? Can you guys tell me what is the function of ribosome again? All right, ribosome is to synthesize protein. Very good. RER is to do what? RER is to transport protein, correct? Golgi Apparatus, give me the first few words enough already, the keywords. What is the 5 alphabet? PMPT, correct? PMPST, process, modify, pack, sort, and transport, PMPST. In your pancreatic cell, we have a lot of ribosome, RER, and Golgi Apparatus. This pancreatic cell, which is your pancreas, why do they have a lot of this? They will produce products of protein, such as enzymes and hormones. Enzymes and hormones, they are made of protein. That's why I say, right, the secretory vesicle has a lot of what? Hormone and enzyme. Because they are the one that secrete hormone and enzyme in your pancreatic cell. We have 3 enzymes in your pancreas. Pancreatic cell is a tissue. Pancreatic cell, they will form into pancreatic tissue. And then they will form into organ, which is your pancreas. So it's the cell organization. Okay, so this enzyme, we have 3. It's called LAT enzyme, LAT. L stands for lipase. A stands for pancreatic amylase. T stands for trypsin. LAT, okay? Lipase, amylase, trypsin. The function of lipase is to break down your lipid into fatty acid and glycerol. Pancreatic amylase is to break down starch into maltose. Trypsin is to break down polypeptide into dipeptide. Okay, so the 3 enzymes. You will learn this in, maybe you already learned it, chapter 4, chapter 5, and chapter 9. Okay, we have 2 hormones here. The 2 hormones are insulin and glucagon. Insulin and glucagon. What is the function of insulin? Is to reduce blood sugar level. Glucagon is to increase blood sugar level. I don't know if you guys heard of people who have diabetic. They have to inject insulin. You guys heard of it before? So, diabetic patient inject insulin is to reduce blood sugar level. Every one of us, when we have no disease, your pancreas can produce it naturally. So when we have food already, let's say you have your lunch already, right? Your body will start to produce insulin. You have to break down, you have to reduce, means you have to convert your sugar into glycogen. When you're very hungry, your body will produce glucagon to produce glucose for you. Every single human, we have insulin and glucagon. Just that people with disease, diabetic mellitus, they have to inject insulin. When they remove the entire pancreas, will the person die? The answer is no, the person will not die. You don't die because when you have no pancreas. The only problem, not cannot say only lah, one of the problem that they will have is they will have diabetic mellitus because they cannot control their blood sugar level anymore. So when you remove, you cannot eat a lot of food, not just fried food. You cannot eat oily food. You cannot eat a lot of carbohydrate because you cannot break down starch into maltose. You cannot eat a lot of protein as well. If you want to eat, you can take the smallest molecule. Means you can take amino acid. Porridge also starch. Smallest molecule is not the sense of the size. It's the sense of the molecular size. Means when you break down. So you can take amino acid. You can take glucose solution. So all of this don't need to digest anymore. What about gallbladder? Gallbladder is for oil, for lipid, because gallbladder is to store bile. Bile is to reduce, bile is to emulsify your fats. So when you have no gallbladder, you don't die also. You just have to reduce the intake of oily food for gallbladder. Nobody will die because of having a removal, okay? Can they still eat lipid? Yes. So they will eat lipid, they will also shit the lipid out. Can you die from being diabetic if it's untreated? Yes. You will get bacterial infection. Usually when you have diabetic, right, you have a lot of wounds. Maybe you fell down, you have wounds, there's bacterial infection, and then when they start to spread your infection, it's either you amputate it or you just wait to die. Infection, right? A lot of diabetic patients, they have difficulty of healing because the blood flow very slow. The blood clotting mechanism is very slow. So people have diabetic, maybe they don't die first, I would say. Diabetic has a stages, not in the book stages, but first, when you have a high sugar level, you cannot heal your wound. Number two, your urine becomes sweet, correct? And then you will get blind also. Because your blood vessel is too hardened, you have no oxygen, so the person will get blind for diabetic patient. If it's very serious, you also get kidney failure. Then eventually you will die. So it's not like diabetic and then die. They have many other complications coming out. How they know that urine is sweet? I'm not asking you to taste the urine, but when you pee the urine, if the ants is going there, and then it means that they are diabetic. If it's not, they can do magnetic tests. Magnetic tests can also show the result from blue to brick red precipitate. Okay, nobody tastes the urine one, like guys. Okay, then okay. Now we're gonna try the question here for organelles. Anybody has question on the notes? Let's try the question together. Okay, let's look at page 1, question 1. Okay, flip to your question. If you have a question, you can ask me, okay? Page 1, not page 1. This is page 6, question 1. Okay, diagram 1 shows an animal cell as seen under electron microscope. We have X, W, Y, and Z. Okay, we start to label X first. X, this is nucleus, and they are not actually sticking to the nucleus. It looks like a Wi-Fi. What is X to you? Very good. This is Golgi Apparatus. Golgi Apparatus. Okay, next we have W. W is a dotted circle. What is W? Correct. Centrioles. Very good. Just like I say, right? 2D diagram, it can be centrioles. Lysosome, usually they will circle the entire. They don't give you dotted most of the time. Lysosome should be a solid circle. If you see a dotted, and also you see a cylindrical shape, the one I showed in the notes for centrioles, then the dotted has to be lysosome. Okay. Why? They are outside of the nucleus. This is nucleus, by the way. Outside of the nucleus, and there is no dot. Actually, why is not a very good diagram. They didn't show whether it is dot or without the dot. We don't know because the diagram is not very clear, right? So we assume that there is no dot lah, okay? This is smooth ER, correct? In a better diagram, they're supposed to draw for you to see whether they got the dot and without the dot. Okay, this is endoplasmic reticulum. But near the surface, actually the dot here, it can be the cytoplasm also. So this is not a very good diagram actually. No, sometimes when we draw cytoplasm, we will also dot the cytoplasm. So it's not a very good diagram. But if it's a good diagram, right, it will be like that. Let me give you another diagram. A good one should be something like that. This also not very good. This one got dot one. Usually on the surface one, you can see this is a rough ER. Okay, now back to this label. What is ZAG? What is ZAG, guys? ZAG is a mitochondria, correct? Mitochondria. Mitochondria. Actually, why? Actually, why, right? It's a rough ER over here. Why? Because later when you do the question, right, they ask you the relationship between X and Y. So next time, I didn't change I forgot to change the question lah. But next time, if you don't know whether it's a rough or smooth ER, you try to see whether the question has, do they ask you anything related to X and Y relationship? If they ask you X-Y relationship, the Y has to be rough ER. Because smooth ER and Golgi Apparatus got no relationship one. So in exam, to confirm your answer, you read the question again. You read all the questions to see whether they ask you, or is there any relationship between X and Y? Then you will know whether Y is a smooth or whether Y is a rough. Must the G in Golgi Apparatus be capitalized? Yes, G has to be capital letter. Okay, assuming that this is Y lah, guys. Okay, assuming that this is Y. Okay, now done already. Next we have B. What is the function of X and Y? Oh, sorry, not X, W and Y. Later we will do a relationship between the Golgi Apparatus, okay? W and Y. W is your centrioles. Centrioles is to form spindle fiber, correct? Form spindle fiber during cell division. Okay, next we have Y. Y is a smooth ER. What is the function of smooth ER? Correct, to carry out detoxification of drugs or to synthesize what? Either one also can, guys. Is to synthesize lipid. Very good. Synthesize lipid. Or you can say to carry out detoxification of drug. Both also correct. Why is the inner membrane of Z folded? Okay, if it's only 1 mark, you only have to write in 1 point. Okay, 1 mark only, right? Anything that is folded in biology, no matter whether they are mitochondria or not mitochondria, right? We have the same answer. Correct is to increase surface area. To increase surface area. Now, if you have to write one more sentence to explain your answer, then you can say that to generate more energy in the form of ATP. One more sentence, you're going to write to generate more energy in the form of ATP by a process. What is a process in mitochondria? To generate energy is by a process called cellular respiration, correct? Cellular respiration. Very good. Cellular respiration. If they ask you to explain your answer, if it's not, actually the first one is enough for you to increase surface area. Cellular respiration is to produce energy from glucose. When you oxidize your glucose, that process is called cellular respiration. Same meaning as aerobic respiration. It's called cellular respiration. Name one example of animal cell that's a lot of components, Zach. What is that again? That is mitochondria. Okay, they ask you to write what? Animal cell. The keyword here is animal cell. We only have two answers. Either you want to write sperm cell or either you want to write muscle cell. Both also correct. So let's say since everybody tell me sperm cell, okay, write sperm cell. Both also correct. Up to you guys. Sperm cell or ovum cell also can. Explain the role of Z in the cell mentioned in D1. What is the function of mitochondria in your sperm cell? They have to generate more energy so that the sperm cell can do what? Correct, can swim. Very good. Swim and penetrate ovum. Very good. Okay, so we're going to say generate more energy to allow sperm to swim and penetrate ovum. When the question has the word called a lot of, your answer needs to write more. Without writing more, your answer is not correct, yeah? A lot, your answer needs to write more. Okay? What will happen if cell in component Z fails to function? When there is no mitochondria, means that the sperm cannot swim. When the sperm cannot swim means that there is no fertilization. If the question says fails to function, then we write the function in front, we add unable. Unable to whatever, okay? Just write unable and then write the function. For all of the question, when they say fails to function, cannot produce, blah, 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 okay? Cannot function on. Unable, and then you write the original function itself. So they unable to swim and penetrate ovum for fertilization. Unable to generate energy in the form of ATP. Okay, so you're going to say unable to generate energy in the form of ATP. Sperm cell unable to swim and penetrate ovum for fertilization. So as you can see, unable, cannot, unable. If the question is 1, 2, 3, then the question is continuous related, yes. When they put A, 1, 2, 3, right, usually they are related. Done? Let me know when you're done with this. We're going to look at E1 already. Okay, next. State one tissue in which in X and Y found abundant. So that's why I say, right, for this question, Y is supposed to be rough ER, but the diagram is not very good. Because Y is supposed to be RER, so that you have a relationship, you have an answer for X and Y. So right now, we only write for X lah. Y we write for different answer later. So X, since this is a Golgi apparatus, which cell has a lot of Golgi apparatus? Correct, your pancreatic tissue. Because it's tissue, right? You're going to write pancreatic tissue. Pancreas is organ. So this is pancreatic tissue. Okay, if they ask you to write X and Y respectively, then your Y is supposed to be what answer? What if it's a smooth ER? It can be found abundantly in where? In your liver cell, correct? Liver cell. If they ask you respectively, okay, it means you're going to write X and Y. Give a reason for your answer for pancreatic tissue. Why your pancreas has a lot of Golgi apparatus? It's because that your pancreas, they are able to produce products of protein. What is the products of protein? We have two examples. Correct. We have enzyme. Very good. The other one is what? Hormones. Correct. Glucagon is a hormone. Glycogen is the storage of food. Yeah. So it can produce products of protein such as enzyme and hormone. Enzyme and hormone. Name one component that only present in plant cell. Only in plant cell is what? Only in plant cell, correct, is chloroplast. And then the other one is cell wall, correct? Chloroplast or cell wall. We cannot write vacuole. Why cannot write vacuole? Because actually animal cell also has vacuole, just that they are smaller. Name one component that only exists in animal cell. Only in animal cell, your answer is centrioles. Very good, centrioles. Okay, done with this, we're going to move to question 2. Anybody has, only centrioles for animal cell, yeah? Anybody has question to ask in question 1? Everybody okay with this? Okay, now then we're going to move to the next question. Diagram below shows the structure of cell observed under an electron microscope. This is a cell. Now, how do I know what is this cell? Is this a plant cell or animal cell? We're going to look at the component inside the cell. Take a smart guess. Is this a plant cell or animal cell? You want a screenshot? Okay. Correct. That is a plant cell. How do you know it's a plant cell? Correct, because they have cell sac. Very good. They have the vacuole, the large vacuole, right? They have cell wall. They have chloroplast. Very good. This is how you look at whether they are plant cell, animal cell. So P, we know that this is a cell wall. Cell wall. R, we know this is a chloroplast. Can you guys tell me what is Q? Very good. Q is the inner layer of cell wall. It's called plasma membrane, correct? Plasma membrane. Okay, it's known as plasma membrane. Yeah, structure P can only be found in plant cell. Name structure P and the function of it. So we know that P is a cell wall. What is the function of cell wall? Is to maintain the shape of a cell. Very good. Okay, so P is cell wall. It provides mechanical support to plant cell. Or you can write to maintain the shape of a cell. Up to you guys. Whichever one also can. In maintain the shape of a cell, can I don't say P is a cell wall? Okay, can you choose not to write this straight away, write cell wall? Are you trying to say this? Okay, this one, actually, I would not suggest you to do it because your answer, most of the time, it has to be in a short sentence. It means that your answer has to be a complete short sentence. You cannot just dash cell wall. In one situation, I can dash cell wall only when they ask you to name without writing the function. So they ask you, name P, and then you can write dash cell wall. But usually, if it's not, then we will write in short sentence. Because some teacher, right, they also like that one. They say that your answer is keyword, they give you wrong. You have to be in a short sentence. You can write dash in exam, guys, don't worry. Dash or dot, up to you. Both also can. As long as your answer is a short sentence. A short sentence, then correct already. No keywords means you cannot write dash support. Okay, dash cell wall. This one definitely cannot lah. Obviously cannot lah. So you have to be in the short sentence, yeah. Q is found in both plant cell and animal cell. Name Q and main characteristic of this structure. Q is plasma membrane. Okay, they are selectively permeable. Very good. Q is plasma membrane. It is selectively permeable. Means we don't write semi-permeable. Okay, we don't write, okay, you can write partially permeable, but selectively permeable is one of the best answer to write. In chapter 3, we also learn plasma membrane, right? In chapter 3, they only want you to write selectively permeable. So write this answer is enough already. What are structure P and Q made of? P is a cell wall. Cell wall made of a sugar that is not sweet. The sugar that is not sweet is called cellulose, correct? So P is made of cellulose. Q is a plasma membrane. Plasma membrane made of, okay, the lipid is not wrong, but make it more complete. It's called phospholipid, correct? Phospholipid and protein. PQ is made of phospholipid and protein. Okay, phospholipid and protein. In chapter 3, we learn what is phospholipid, right? It looks something like that. I can only use my maximum drawing to draw for you to see, guys, okay? This is phospholipid. And then in between, maybe we have a channel protein, something like that, okay? So this is phospholipid and protein. A bit ugly, but yes, it looks something like that. Max, I can only draw this, okay, guys. If I write phospholipid bilayer, actually also correct. Okay, next, state two example of the cell sap content. What is inside the cell sap? Very good. Nutrients. You can also write. All right. You can also write water. You can also write. What else, guys? Water, nutrients, waste substances. You can write glucose, amino acid, gases. There's a lot of things for you to write. Amino acid, glucose, water, mineral, gases. Any two, up to you. What? Somebody asked me what? Is cellulose tasty? You also eat cellulose, so don't worry. Every day you're eating. When you're eating any form of vegetables, you're eating the cellulose already. Dissolved gases is a better answer. Yes, okay. Dissolved gases, yeah. Lysosome in animal cell. Actually, yes, lysosome only can be found in animal cell. Plant cell don't have lysosome. but they don't write it in the textbook. If I don't remember, they write it in textbook, okay? Describe the effect of absence of cell set in herbaceous plant. Herbaceous plant is what? Herbaceous plant is non-woody plant. Herbaceous plant is non-woody plant, okay? It's called herbaceous plant. Absence of cell set. When there is no cell set, what cannot be maintained? Correct. No turgidity. Very good. No support. No turgidity. Unable to maintain turgor pressure. No turgidity. Unable to store your cell set. No nutrients, no glucose, no water. Nothing. Okay. No support at all. So we're going to say herbaceous plants loses its turgidity. Plant cell unable to store nutrients, or you can write water or dissolved gases. Or you can say unable to provide support also can. So again, the question says absent. So you're going to write negative answer. Unable to maintain turgidity also correct. Loses its turgidity also correct. Okay. Read the following information. Structure R is usually found on the cell of green plants. Green plants is the keyword, like green plants. They have to be green color. Based on statement above, explain the importance of chloroplast to green plants. Chloroplast contains of green pigment. The green pigment known as chlorophyll, correct. Chlorophyll function is used to trap light, correct. Trap sunlight and convert into chemical energy during photosynthesis. Very good. Okay, in 2 sentences, we're going to say chloroplast. Or you can write structure R if you want to. Contains of chlorophyll. Chlorophyll trap sunlight and convert into chemical energy during photosynthesis. And convert into chemical energy during photosynthesis. So not chloroplast trap sunlight. It's the chlorophyll that traps sunlight. So we cannot say chloroplast trap sunlight. If you write chloroplast trap sunlight, your answer is wrong. It has to be chlorophyll trap sunlight. Okay, write one word equation to show overall reaction that occurs in organelle R. Word equation. What does word equation here mean? It means you cannot write O2, CO2, H2O. We have to write water, carbon dioxide, oxygen. You cannot write chemical formula, but don't need to put it into a sentence. It's like an equation. To carry out photosynthesis, what do they need? Number 1, they need carbon dioxide, right? CO2, right? So you're going to write carbon dioxide. Not CO2, but carbon dioxide. And they also need water. Very good. Plus water in the presence of 2 things. Photosynthesis, obviously they need what? Sunlight. Light is being trapped by chlorophyll. Very good. The top part, we write sunlight because sunlight is on top, right? Sunlight. They produce into 2 products. The 2 products will be, correct, glucose and oxygen. Because in Form 4, we don't learn they produces water, but this is in Form 4 syllabus. No energy. Don't have to write energy. In equation, there's no need to write energy. The chemical energy is the glucose. So don't need to write the energy anymore. The chemical energy is actually the glucose. Okay, next. So this is a word equation, guys. Okay, next. State 2 types of tissue where organelle R can be found in large number. Organelle R is chloroplast. Chloroplast can be found abundantly in which cell? Very good. Okay, we have palisade mesophyll cell. The first choice that you have to write is always palisade mesophyll cell. Palisade mesophyll cell. Okay, second, we can have two more, which is spongy mesophyll cell. Very good. Spongy mesophyll cell. Or you can write gut cell. Even though spongy mesophyll cell and gut cell, they don't have a lot of it, but we can still write because we have no other answer to write already. Or you can write, yeah, this is tissue. Yes, you can write tissue. Palisade mesophyll tissue. Thanks. I didn't read the question just now. It's supposed to be tissue. Okay, tissue, yeah. Okay, justify ribosome. Why ribosome is attached onto the rough ER. So this is relationship. They ask you, why do you think ribosome need to go onto rough ER? We talk about what? The function of it. We start with ribosome. Ribosome is to synthesize protein. Very good, synthesize protein. Why they have to attach onto the rough ER? Because rough ER helps them to transport protein. To where? Transport protein to? Very good, transport protein to the Golgi apparatus. So 2 marks means you write these two functions. First, you're going to say ribosome. Synthesize protein. Protein is needed to attach on rough endoplasmic reticulum to attach on rough endoplasmic reticulum to transport synthesize protein to Golgi apparatus. Do we need to say the both function of rough ER Golgi apparatus? Then we write ribosome synthesize protein and then the rough ER transport protein. It's already in the sentence. So your answer, you need to write the function. No matter how you form your sentence, but you have to bring the function into your sentence. Okay, to complete it, yes, you can write the vesicle. Okay, which vesicle transport to the Golgi apparatus? Through or via also can. Via means through, yeah. Correct, the transport vesicle. The transport vesicle. To make it more complete, we're going to write transport vesicle. Usually when we write transport vesicle, it's for your Form 4 Chapter 5. Now with this, done, let me know. We're going to move to the next question. Wait for 5 of you. We're going to move to the next I. Can you write synthesize or mean with the Z or with the S? Is it actually can both also can, just that one is UK, one is US. In biology, they don't really care, I would say. Because I don't know whether in English your teacher can or not, but in biology, they don't really care, okay? 2.3 shows the longitudinal section of an onion bulb. The onion bulb is planted in the soil. Onion bulb. What color, guys? Onion is a plant cell, right? What is the color of onion? Onion is never green in color. They are purple, right? They are not green in color. So, do you think onion has chloroplast? No chloroplast, but they are still plant cells. They have no chloroplast. What is the function of this onion bulb there? This onion bulb is to store food. It's to store nutrients. When they store nutrients, they are underground, right? It will help the plant to germinate. Germinate means form into leaves. It's called germinate. So they will use the food to form into leaves. The spring onion come from this. That's a spring onion. So the food inside, they will help to germinate, to form into leaf. So in the soil, the onion scale actually they will shrink in size. They will grow slower in the soil because the food is being used up in the onion bulb itself. To write in sentence, they ask you, explain why organelle R is not present in onion scale leaf. Germinate means they form into leaf. It's called germinate. They form into leaf. When the seed form into leaf, it's called germination. Okay, they ask you why don't have, because they don't have to carry out photosynthesis. So you're gonna say, onion scale leaf does not carry out photosynthesis. It is used to store food. It is used to store food. Okay, explain what happens to the growth of an onion plant if the onion bulb is placed in the dark cardboard. They ask you, growth. Okay, I mean, we all understand that they don't need to have sunlight because they don't carry out photosynthesis. But the growth of onion, when they are under the soil, just as I mentioned, right, they will use up the food to germinate. Will the growth become faster or slower to the onion bulb? Slower. Why? Because the food is being used up, right? It will be slower, right? Because they have to form leaf. But in the dark or without the dark, it doesn't cause any difference because they don't need to have sunlight. This is just to trick you only, guys. When they say place in the dark cupboard, it's just to trick you. I mean, not related at all because they don't have to use sunlight, right? So we're going to say the growth become slower. This is because food storage in onion bulb is used to form new leaves. It will not remain the same unless they don't germinate. If they don't germinate, maybe they will remain the same. If it's not, it becomes slower. The graph of a plant cell, you will learn it in Form 5. Okay, it looks something like that. I can give you an idea of it. So initially, they will decrease and then they will increase again. So the decrease point here is during germination. Increase here because they already have leaf. So they can carry out photosynthesis. How to get more food? When they germinate, they have leaf already, right? They will increase in growth. Only when they start to form leaf. All onion will only form leaf. Okay? So this is the graph of it. They will reduce and increase later on. Okay, done with this? Okay, can I say it does not have chlorophyll for eye? You mean one of the point, is it? Because it already says that there is no organelle R. So when you say it does not have chlorophyll, means you are kind of repeating the question. Okay, Ken? Okay, now next, we're going to look at question 3. Diagram 16 shows the structure of plant cell wheel under electron microscope. Label P, S, T, U, V, W. Okay, label everything together. Label P first. They say this is a plant cell, obviously. Outer layer of plant cell is what? Outer layer of plant cell is called cell wall. Very good, cell wall. And then for S, it looks like a Baymax eyes. What is S? S is a chloroplast, correct? Chloroplast. And then we have T, one large vacuole. All right, one large vacuole. U looks like a Wi-Fi. What is this Wi-Fi thing here? Golgi Apparatus. G has to be capital letter, yeah? Golgi Apparatus. V, it looks like a cloud inside. What is V? V is a mitochondria, correct? Mitochondria. W is this entire thing here. What is this entire thing here? W is nucleus, correct? Nucleus, W is nucleus, yeah? So A already labeled A1, A2. State the function of following structure S, U, and V. S start with chloroplast first. What is the function of chloroplast? If you have to only use one sentence to write, if you don't want to write contents of chlorophyll, trap sunlight, convert into chemical energy, the one short sentence to write the function of S is carry out photosynthesis. That's the shortest answer for you to write. So S, carry out photosynthesis. And then we have U. U is Golgi Apparatus. This one you cannot short form already because we only have 5 words to write. Correct. P, M, P, S, T. Of course, we cannot write short form, right? The first P is what? What does the first P stands for? Correct. The first P is process. The first one is process. M is Modify, very good. And then next P, pack, correct. Package, or you can write pack, also can. Or you can write sort. The last one is transport, very good. What do they modify, pack, sort, and transport is actually your protein. If it's only one sentence, just write this one. You can write protein, you can write carbohydrate, you can write glycoprotein. Okay, W. What is W? W is your nucleus. What is the function of nucleus? Nucleus contains of DNA, correct? Or you can write control all cell activity. This is also a correct answer, yeah? It contains of DNA that determine characteristic of cell. I will write the one that I say. If you want to write control all cell activity is correct. Contains of DNA that determine characteristic of cell. Or you can say control all cell activity, up to you. State one similarity between S and B. S is chloroplast, V is mitochondria. We only have one answer to write. Similarity, very good. Not energy, guys. Just like I say, you can write energy, right? You can only write both have double membrane. Only one answer to this. Both have double membrane. Both have double membrane. Can I just label the name for in diagram for A1? Cannot. You have to write it over here in exam. Okay. You cannot write it over here, yeah? You're gonna write it in the space given to you. Can I write made of double membrane? Can. Okay. Explain why structure V has a folded inner membrane. We write the same answer just now, right? As long as they are folded, is to increase surface area. Increase surface area. Next, to generate more energy in the form of ATP. Okay, again, some of your teacher is very specific. Actually, this is also correct, but the some of your teacher is very specific. They must, they want what? They want you to write the starting who. Means you're gonna say inner membrane to increase surface area. Or at least you're gonna write this is to increase surface area. So some teacher, they are very specific over this thing. You're gonna write this is to increase surface area. Without writing this, your answer is wrong. Okay, to generate energy in the form of ATP, you're gonna write by cellular respiration. By cellular respiration. Okay, explain how the compound in structure P helps to carry out function. P is a cell wall. P is a cell wall. Can you guys tell me, cell wall has what characteristic to allow them to carry out function? If you write cellulose fiber, you have nothing to write anymore. Because you write, P is made of cellulose fiber. But the function that you know, for example, you know the function is to maintain the shape of cell wall, right? Doesn't match with the characteristic. So your characteristic is to write tough and rigid, not turgid, it's rigid. It is, or P is tough and rigid. This is to maintain the shape of cell. Tough and rigid. So this is the best answer to write. If you want to write cell related, you always write this. You can write strong and rigid lah. Either you write strong or tough, but you must write the word rigid. Anyhow, the rigid word is a compulsory word. You have to write the word rigid. Okay, next, state two types of cell in which B is found abundantly. You can write animal cell, you can write plant cell. So how do we want to draw? What is the answer for animal cell and plant cell? Correct. You can write sperm cell, we can write muscle cell, we can write one more for plant cell. What is a plant cell one called? Very good. Meristematic cell. Meristematic cell. Meristem cell and meristematic cell is the same thing. Up to you which one you want to write also can. Draw structure B. Draw a mitochondria. We're gonna draw two layer. Oval lah. Okay, one more layer. Okay. And then we're gonna draw sorry, wrong one. Not two layer. The two layer is a cloud shape. Wrong diagram. It's supposed to be like that. No, this is mitochondria, not chloroplast. This one I don't want to draw chloroplast. This is mitochondria. Then we have to write this is outer membrane. This is inner membrane. It is outer membrane, inner membrane. No need to circle because double membrane they are referring to. This is the first membrane. This is the second membrane. Got no two circle one. Two circle one is chloroplast. Would it ask in exam? Sometimes they will ask in exam. What is within the two membrane? Actually, they have other labeling that we don't learn. They have a thing called matrix and crystal, but we don't learn the content inside. Okay, predict what happens if plant cell, if V is absent. Keyword is plant cell. When there is no plant cell, what will happen? When there is no mitochondria, what happened to plant cell? It means you have to write what? The meristematic cell, right? Because it's the one that can be found in plant cell. What happened to meristematic cell? Unable to, correct, unable to grow, unable to carry out cell division, unable to produce new cell to carry out cell division. Your first sentence, you always write unable to generate energy first. Unable to generate energy in the form of ATP. Next, you're gonna write meristematic cell unable to carry out cell division to produce new cell. So always remember, absence, we have what? Unable, cannot of. Exam drawing need to use pencil. Label, you can use pen. All drawing in biology is pencil. Done? Let me know when you're done already. Meristematic cell is a young plant cell. Okay, what are the characteristics which enable you to differentiate plant cell from animal cell? For structure question, you cannot draw in table unless the teacher allow you. But you know, you don't draw table in exam. We only draw table in essay question. Structure question, you have to form sentence. How to form sentence? Tell me one differences between animal cell and plant cell first. Plant cell has what? Animal cell don't have. Cell wall. Okay, let's say the cell wall first. So you're gonna say plant cell has cell wall, whereas animal cell does not have cell wall. Your answer needs to use whereas or while. Got one student use but lah, but the teacher give him wrong. I don't know why. So if your teacher say cannot use but, you use while and whereas. Okay, next, chloroplast. Okay, right, chloroplast. Plant cell has chloroplast, whereas animal cell does not have chloroplast. What animal cell has? Centrioles, correct. Animal cell has centrioles. Whereas plant cell does not have centrioles. Random question, but can use because in question. I remember last time is cannot one, but I've seen multiple. Because what? You can write because. This is because of course, can write because. Why cannot write because? Use because in sentence. Plant cell has cell wall because, something like that. Are you trying to say this or what? Usually because is to explain your statement. You can write because in biology. For example, okay, this is because, yes. Fixed shape and irregular in shape is etc. Yes, correct. Okay, now, in one school, okay, in one particular school, the teacher says, cannot write animal cell has no fixed shape. The teacher want them to write irregular in shape. So if your teacher is so specific over this thing, then you don't write plant cell has a fixed shape, whereas animal cell has no fixed shape. You're supposed to write plant cell has a fixed shape, whereas animal cell has irregular in shape. Okay? Last time is cannot use because. Can write in explanation, we can write because. Like just now, for example, mitochondria, right? This is to increase surface area. To increase surface area, right? Then you can write this is because energy can be generated more, something like that. You can write because in exam one for biology lah. But okay, I will say only some teachers are so specific over small thing. Can you write plant cell don't have lysosome? Yes, you can. Up to you. In table, you can write it in essay. If your exam is having full paper, maybe this is your first exam, you don't know about your exam format. If your exam is full paper means what? Let's say your midterm is 2.5 hours for paper 2, it will be a full paper. So paper 2 actually has section A, B, and C. Okay, if your exam is 2.5 hours 2 hours 30 minutes, a full paper. Section A, we have 8 structure question. All question you have to do it. Section B is 2 choose 1. Essay. Section C, only 1 essay you have to do it. So in total, you have to do 10 questions. 8 structure and 2 essay. B is 2 choose 1. No need to do 2, just do 1 question enough already. Our teacher won't mark for you guys. Section C is not experiment. Section C is section B. Section B and C is the same thing, just that B you can choose, C you cannot choose. Okay, they will convert back into 100 marks, yeah? So, section A, 8 question is 66778899. So the question is like 1, 2, 3, 4, 5, 6, 7, 8, right? So it's like 2, 6 marks, 7 marks, 8 marks, 9 marks. So, I don't know whether your first term exam is full paper or not. I mean, I know some of the teacher is not having full paper. This is 20 marks, 20 marks. If I screw this paper, it's over. No lah, won't want, guys. Really won't want. Your first term exam, it will not be as difficult as what you think it is, as long as you study. In exam, essay must write in table or you can write in paragraph, guys. In essay, you can write in like that. But table is faster lah for essay. I mean, if you write animal cell, plant cell, then you write presence of centriole. This one, absence of centrioles. You can write in table. Table also need to use short sentence, yes. Can every essay use table? Only differences you can use table. No differences how to write in table. The only differences can write table, guys. Okay, now it is, we're going to go through the next part, which is amoeba species and paramecium species. So now that are you guys confident to answer your cellular component already? Okay, confirm can get 100 marks already, right, for this part. Easy, okay. Now, by the way, maybe some of you don't know how to label this diagram because you never see this, right? We just label, but we don't do this question. We just label this question together. For P, is also one of the organelle. So P, right, if you look like a cheese diagram, this is a smooth ER. I reckon in short form, just for you to know, guys, okay? And then for R, there is this vesicle here. It looks like a Wi-Fi. This Wi-Fi here, Q is your Golgi Apparatus. R is your rough ER. RER, okay, rough endoplasmic reticulum. S, this is a hydrolytic enzyme. So obviously this is a lysosome. T, this one quite obvious. You know what is T already. T is centrioles, correct? U is mitochondria. V is a nucleus. Actually, when you know the labeling, then you should know the answer. So hopefully in exam, if they come up with this part, you can get full marks. The one that we are trying is all exam type question. Okay, now then let's look back to your notes. We're going to do amoeba and paramecium species right now. So amoeba and paramecium species, actually, like I want to say, today I cannot finish page 4 and page 5. It's not possible for me to finish it. Page 4 and page 5, you can look back to my YouTube video. I teach 4 and 5, I think 2 years ago. You can find the video 2 years ago, okay? So I will only teach, up to amoeba and paramecium species today because no time, guys. Amoeba and paramecium species, the first thing you have to know is the name. A capital letter, P capital letter. Remember, A capital letter, P capital letter. Yeah, it's a fixed thing. You cannot just write small letter. Amoeba species has no fixed shape. It looks like a jelly, no fixed shape. Paramecium species looks like a slipper, so it has a fixed shape. Amoeba species start from the labeling. The outer layer of the amoeba species or even paramecium species is known as plasma membrane. Don't need to write this. Just write plasma membrane. Plasma membrane. So gases dissolve through the plasma membrane. Okay, water goes in through here as well. Everything goes through the plasma membrane. Inside has three things. We have food vacuole, the one with food, like stomach, the one with food. Contractile vacuole, there is empty. Okay, and then we have nucleus, the one that is colored. So when you see three in exam, because it's black and white, right? Food vacuole, they must have food inside. Contractile vacuole is empty inside. And then nucleus, it has to be fully shaded. This is how we differentiate these three things, yeah? On the hands, okay, they will bracket this thing, they will ask. On the hands, it's called pseudopodium. Pseudopodium, it can be the hand, it can be the leg. It's called false limb. They are known as false. False is not real, it's false limb. Limb referring to hands or leg is called false limb. They are able to move by using pseudopodium itself. This is the labeling for amoeba species. For paramecium species, you got more things to label. They have a fixed shape. The outer layer, we also have plasma membrane, but they have this hair-like structure on the outside. It's called cilia. It's cilia. Okay, cilia is the hair-like structure. It's for them to move. It's called beating of cilia. So they'll move like that. It's called beating of cilia. The mouth is known as oral groove. Oral means what? There's a brand called Oral-B, right? Oral, it means the mouth. Groove is the one that went in. It's a groove. Groove is a thing that it goes in. It's called a groove. In English, it has the same meaning, right? It's called groove. And then we also have contractile vacuole. This contractile vacuole, it looks like a sun. Sometimes looks like a star. Don't have to write anterior, posterior. You only have to write contractile vacuole. Don't need to write anterior, posterior. Anterior means the front, posterior means the back. That's called anterior, posterior. And then we have the nucleus. We have big and small nucleus. The big nucleus is called macronucleus, the big one. The small one is called micronucleus. How to know which one is big or small? Remember Big Mac. You know, Big Mac is big, right? MAC is big, right? Big one is called macronucleus. In your textbook, generally, they're only known as nucleus. Why do they need two nucleus? Because the small nucleus helps in reproduction. Okay, they will help in reproduction. The big nucleus, they will control all cell activity. So they have two nucleus. But generally in your textbook right now, they only write nucleus. But they have two different functions. One is to reproduce, one is to control all cell activity. And they have a butthole. The butthole is called anus. Anus can be any part. It can be here, it can be here, it can be here, it can be any part, as long as their hole is called anus. So they use their mouth to eat. They form into a food vacuole. The anus will remove the undigested food. This is the labeling for paramecium species. In biology, first thing that you learn, you need to start from labeling, guys. If you cannot label the diagram, it means you don't know the answer anymore. You cannot write the function. You cannot write the process. So whenever you learn biology, you must learn the labeling first. Hey guys, you cannot write asshole, please. Anus. Okay. Amoeba species and paramecium species both can reproduce. We have sexual and asexual, right? Okay, what does sexual and asexual means? A means absence. Absence of sexual partner means me, myself, and I is called asexual. Sexual means what? There is a partner. It requires two for them to reproduce. In amoeba species, it's only asexual. It's just me, myself, and I. In favorable condition, how do we define as favorable? These two species, they live in water. They live in lake, they live in the pond, they live in waterfall. All of these are called fresh water. In favorable, enough of water, enough of oxygen, enough of food, they will carry out binary fission. 1:2 is called binary fission, 1:2. And if it's not favorable condition, they will form spore. Like mushroom is called formation of spore. How do we define as not favorable? When they are dry, when they are in hot temperature, when there is no food, no gases, it's called not favorable. So favorable, they will split lah. Not favorable, they will just form spore. Paramecium species. Over here, sexual and asexual, right? In favorable, same thing, they will split themselves, 1:2, 2:4, split themselves, everything is same. And then for sexual, they have conjugation. What is conjugation? Conjugation here is where two paramecium species, the mouth and the mouth, like that, okay? They will face together and they will exchange the nucleus, which is the micronucleus. When they exchange, both will also have baby. In another word, they kiss, then they got baby already. Only paramecium species can kiss, have baby, okay? They kiss, they put their mouth together, right? They exchange the nucleus, right? Both will have babies. This is what we call as conjugation, guys. Conjugation. Don't write kiss, have baby, okay? Please you all, okay? Conjugation, not favorable. When they are dry, they will just kiss and have baby, like conjugation. Now, amoeba species, how do they eat? They always ask in the exam. The feeding process of amoeba species is a very common asked question. Yeah, they exchange the nucleus, correct? They exchange the micronucleus. Okay. Okay. How much you have heard this is still very good. Okay, well, they kiss, they have baby. You cannot see them also, they're in the water. The eating process. Conjugation produces two paramecium species. No, they will release egg kind of thing. They will form into paramecium species. It could be more than two. Only the micro, because micro helps in reproduction, right? Only the micro one. The macro no need. Only the micro one. The small one. Okay, now feeding process. Phagocytosis is an eating process. How amoeba species eat? Imagine I am amoeba species. Okay, this is the food. Okay, I'm eating a food. They eat bacteria most of the time. So in their life, they have to go to the food. Their food don't go to them because they got no grab food, guys. Okay, they have to go to the food, man. They have to approach the food by extending the pseudopodium. The pseudopodium will extend and then they will engulf them. Engulf means what? They hug the food. Okay? Engulf means they surround the food. Like it's called engulf the food. They form into a thing called food vacuole, which is the one that we learned just now, food vacuole. Okay, they form into food vacuole, and the food vacuole has to digest the food, right? How do they digest the food? They will fuse with lysosome. Can you guys tell me again what is in the lysosome? We have lysozyme, correct? The lysosome, secrete lysozyme, they will digest the food. The food is being digested. The nutrients are absorbed into the cytoplasm. They absorb nutrients for what? They will go through a process called assimilation. Assimilation means they will use the food. Why do they need to have glucose? Because they need to generate energy. It's called assimilation. Why do they need to take protein? Because they need to form new cell. The process is called assimilation. Human, we also have assimilation, guys. Means you use your food to something else. It's called assimilation. Okay, when you eat the food already, absorb the nutrients. Okay, now we have shit, right? We don't call shit in biology. We call undigested food. Undigested food, it will be left behind as amoeba species move forward. So any part of their body can be the mouth. Any part of the body can be their anus. No specific area, it has to be the mouth or anus. So they'll just move to the food. They like food, they go to the food. Okay, so this is feeding process. It's called phagocytosis. For paramecium species, it's a bit different because paramecium species uses the cilia to move. It's called beating of cilia. The food enter to the mouth. They form into a food vacuole. The lysosome fused with the food vacuole, secrete enzyme, absorb nutrients. Because they have anus, right? The undigested food will go out from the anus. So they have a specific area to remove the undigested food, which is the anus. So there's difference between these two, yeah? This is for amoeba. This is for paramecium species. Most of the time, they ask about amoeba. Most of the time, I would say, out of 10 exam paper, 8 exam paper, they ask about amoeba. Why? Because in next few chapter, you still have to learn what is phagocytosis. Okay, next one, excretion is also a very common asked question. No matter whether it's amoeba or paramecium species, both have the same answer. Contractile vacuole is involved in excretion process. They will go through a process called osmoregulation. Osmo means water. It's called osmo. Regulation means regulate lah. Okay, like control is called regulate. When the term ends with ion, it means it's a process. It's a process of controlling water balance in the cell. It's called osmoregulation. Okay, controlling water balance in the cell. As I've mentioned, this lives in fresh water. Imagine amoeba species and a fresh water pond. Okay, which one has more water? Of course, the fresh water, right? So the water will keep on going to the cell. Even going in. When they reach a maximum point, they have to release the water. If they don't release the water, they will die. It's like human. If one day you drink 5 liters of water, you have to pee 10 times, for example. You drink 1 liter of water, you pee 2 times. It means what? More water you drink, it means more time you pee. More water it goes in, more water they will go out. Makes sense, right? If the water keep going in, like human, we keep on drinking water without peeing. Number one, you'll pee on your pants. Number two, you get kidney stone. For them, when the water keep on going in, right, they cannot remove the water out. Then the cell will burst. It's a bursting of cell. Okay, so but we cannot see them kaboom lah. There's no explosion. It's a bursting of cell. Should tell why your school teacher don't let you to go to pee? Is it so sad one, man? It's not supposed to hold your pee, man. You might get UTI also, not just kidney stone. You get infection also. I don't know. Maybe because that you keep on going toilet from the same teacher. I don't know lah. Bursting of cell of what? Lysis of the entire cell. The entire cell will burst because this is unicellular. There are only one cell. The entire cell will just burst. It's just lysis. Okay, you just break down lah. So when water goes into the maximum size, expand to the maximum size, they will contract and expel water out. Every single second, they will have this process. Every single second, they will have this process. Okay, so this is excretion process. Very important. Feeding process, very commonly asked. Excretion process, also very commonly asked. And labeling question lah. You know, done with this, we're going to try some question right now. We're going to try amoeba and paramecium species. Anybody has question to ask in the notes? Okay, no, let's look at the question. Let's look at page 19, question 7. Page 19, question 7, guys. Page 19, question 7. Diagram below shows a unicellular organism. Again, what does uni means? Uni means one cell. They are only made of one cell. It's called unicellular organism. Let's label what is X, Y, Z. The diagram here shows what species before we label X, Y, Z. The species here is what species? This is? Correct. The diagram here shows an amoeba species, right? This diagram here shows amoeba species. Okay, label X, Y, Z. X is empty inside. There's nothing inside in X. So X is a contractile vacuole. Very good. This is contractile vacuole. Contractile vacuole. What about Y? Can you see this shaded, but it's not fully shaded? It's like half shaded. Got food inside. This is a food vacuole. Okay, and then Z, they bracket the hands and the leg. It's called pseudopodium, correct? Pseudopodium, okay? And this is a nucleus lah. This is a nucleus, okay? So we have the answer for X, Y, Z already. Remember in exam, don't label in the diagram. I'm going to label here. What is label Z? Okay, because we have label Z, I forgot to change the question. It's supposed to be, what is the function part label Z? Function. What is the function part label Z? For movement, we call this as locomotion. Locomotion and movement is the same thing. For locomotion. If you want to talk about the specific thing between locomotion and movement, locomotion is to move from one point to another point. We call this as locomotion. Movement is like me right now writing, only moving my upper body is called movement. Locomotion, you have to change in different, like me walking to the door, it's called locomotion. So this is for locomotion. For feeding process also can, but the best word is locomotion. Feeding process is not wrong actually. Usually we write locomotion because feeding process means you have to move to the point, right? You don't move to the food also, right? So it's under, I mean like it's have to move. Explain how organism in diagram obtain food. When the question is more than 4 marks, if it's 5 marks, it should be section B and C already. Because section A question will never be more than 4 marks. So if it's 4 marks and above, it has to be B and C. It's like short essay question. The eating process of amoeba species is known as, what is the eating process of amoeba species called? Correct, it is known as phagocytosis. Very good, okay. You go to the food or the food goes to you? Which one? I go to the food, right? Okay, you go to the food, right? You go to the food. You extend the pseudopodium and you use the word engulf. Engulf is the word, yeah, ENGULF. Engulf means you surround the food. They form into a food vacuole. This food vacuole has to fuse with, when they surround it, they fuse with what? They fuse with lysosome. Very good. Lysosome secrete lysozyme. Digest the food, absorb the nutrients, assimilated. Undigested food, it will be left behind as the amoeba species move forward. So your answer will be amoeba species carry out phagocytosis. Next, we're going to say amoeba species approach and engulf food by extending its pseudopodium. Food vacuole is formed and fused with lysosome. Lysosome secrete means they produce a secrete lysozyme to digest food. Nutrients are absorbed into cytoplasm and being assimilated. Last sentence, undigested food is left behind as amoeba species moves forward. Assimilated means you change your glucose into energy. You change your protein into amino acid. Means you use the food to something else. Like human, we have fats, right? We eat lipid, right? Is to act as a heat insulator. That is also assimilation. Okay, wait for 7 of you to finish this. Let me know when you're done with this, okay? 1, 2, 3, 4, 5, 6, 7. Okay, next question. Done already, yeah? Okay, next one. State the method of reproduction of cell in dry and cold environment. Dry and cold environment. Okay, what is the type of reproduction? Not favorable. Okay, so how do they reproduce in not favorable condition? Correct. Not favorable for amoeba species. It has to be spore formation. They are under asexual. Okay, it's just me, myself, and I. Right. So it's formation of spore. Formation of spore, formation of spore. Only Paramecium species has sexual. State the habitat where this organism can be found. They are found in water. We call this as fresh water. We don't write water, but write fresh water. It's called fresh water pond. As long as they are fresh water, you know we have seawater, right? Seawater is a type of water. Fresh water is a type of water, right? So it's called fresh water. How osmoregulation occurs in this organism? All of this process occurs through which part? The contractile vacuole. If you compare fresh water and amoeba species, who has more water? Is the fresh water or is it the amoeba? Of course, the fresh water. In chapter 3, we learned two new words in chapter 3, right? We learned a word called hypotonic and hypertonic. Hypo means what? More water. Hyper means what? Lesser water. Water always go from more to less water. It's always hypo to hyper. More water to lesser water. Your fresh water is always hypo. When water diffuses in, when they reach a maximum size, they will contract and expel water out. In 3 sentences, first we're going to say, fresh water pond is hypotonic to is what? Amoeba species. Okay, next we're going to say water diffuses into contractile vacuole of amoeba species by osmosis. Any diffusion, we have to write the process. Gases, we have to write simple diffusion. Water, we have to write osmosis. Then you're going to say, when contractile vacuole expand to its maximum size, it contracts and expel water out. It contract and expel water out. Then let me know, guys. Wait for 5 of you, okay? 1, 2, 3. 3, 4, 5. Okay. Next, explain how gases exchange occurs in this organism. Gases exchange. When you see the word gases exchange, they must involve 2 gases, which it will be oxygen and carbon dioxide. Gases exchange is always oxygen and carbon dioxide. And because they are also under passive transport, it's always more water to lesser water. More water. More oxygen to lesser oxygen. It's always simple diffusion. More oxygen to lesser oxygen. More carbon dioxide to lesser carbon dioxide. It's always this. It's always more to lesser, yeah? To explain this, we have to mention the process is called simple diffusion. More to lesser oxygen, more carbon dioxide to lesser. Every organism wants oxygen, so oxygen goes in. Carbon dioxide goes out. In sentence, we have to say, gases exchange occurs through simple diffusion. Okay, in gases exchange by moving down the concentration gradient. Yes, you can write this. In gases, if it's in human, right, it has to be very specific word. We cannot write concentration. We have to write partial pressure. Some teacher also mentioned that you have to write partial pressure in organism, which is this. If it's not, you can write concentration. Okay, I'll write partial pressure. I'll explain to you what is that. So you're going to say higher partial pressure of oxygen in surrounding water causes oxygen to diffuse into amoeba species. Next, higher partial pressure of carbon dioxide in cell causes carbon dioxide to diffuse out from amoeba species. So the word partial pressure here, it means concentration. So you have to remember when we talk about gases, we have to write partial pressure. When you talk about water, we have to write water potential. The rest of the thing, you can write concentration. If it's your exam, is chapter 3 also lah, okay? Water, you have to write water potential. Gases, we have to write partial pressure. The rest, you can write concentration. So only two things is specific, which is gases and water. Don't use the wrong word, guys. Wrong word, your answer is also wrong. For water, use water potential. Higher water potential to lower water potential. Okay, done? Sucrose, you can write concentration. Okay, one more question. We're going to do paramecium species right now. Okay, done already? We do paramecium species, yeah? Everybody done? Wait for 5. Oh, you done? 1, 2, 3, 4, 5. Okay, now next. Okay. Next one, we look at question 8. Diagram below shows an organism which make up of only one cell, which is also unicellular. We label V, W, X, Y, Z lah. We start with V first. Even writing down the concentration cannot be accepted as well. Writing down the concentration gradient is correct. If you write oxygen move down the concentration gradient, is actually correct. But if you talk about gases exchange, you anyhow, you have to explain your answer. Because sometimes if the question is 3 marks, you need to write oxygen go where, carbon dioxide go where. So your answer, if you only write that one, is your first mark. Then you have second, third sentence. So you anyhow, you still have to talk about your oxygen or carbon dioxide lah. But it's not wrong to write move down the concentration gradient. It's correct to write that. Okay, now V, W, X, Y, Z. We start with V. V is bigger one, right? We have two things together here. We have big nucleus and small nucleus. What is the big nucleus called again? Big nucleus is called macronucleus, correct? In exam, you can just write nucleus because your textbook only write nucleus. Okay, called macronucleus. Okay, W is what? It's the oral groove. Very good. This is the mouth oral groove. X is the hair-like structure called cilia. Very good. And then Y is the butthole, guys. It's anus. Z looks like a star, like a sun. These two is the same thing, by the way. This and this is the same thing. All right, it's called contractile vacuole. Contractile vacuole. What is this organism? This organism looks like a slipper. We call this as paramecium species. Very good. Paramecium species. SP dot. You can write SP dot. Or you can spell the word species if you want to, but SP dot is good enough already. B, we have the answer already. What is the organelle below the nucleus? This is the vacuole actually. The small, actually it's supposed to be food vacuole lah. But there's no food inside right now. Okay, next. Can you write SP dot in exam? Yes, you can. You can write SP dot over here. State the function of structure V. What is structure V here? Structure V, nucleus function. The big nucleus, if you don't know what to write, right, just write a normal function of nucleus, which is to control all cell activity. Is to control all cell activities. Next, what is the function of the part labeled Z? Labeled Z is contractile vacuole. To use one sentence to write the function of contractile vacuole is the process. Can you tell me the process of contractile vacuole to control water balance in the cell? The word used is known as osmoregulation. Very good. Osmoregulation. So we're going to say to control osmoregulation, or you can say involved in osmoregulation or helps in osmoregulation, also correct, up to you. You can choose control, help, or aid, or involve. As long as you write osmoregulation lah. To carry out osmoregulation also can. Explain how the organism can survive in the fresh water if it's hypotonic to the cytoplasm of organism. The question is the same, 3 marks, right? But because just now in amoeba species, the first mark that we write is the question, hypotonic. So we have to replace the first sentence right now. We're going to replace with the process. The process is osmoregulation, right? So your first process is Paramecium species carry out osmoregulation. The next 2 sentences is the same as this one. This one and this one, same. Yes. No matter amoeba, paramecium species, it's the same. Can you all write this by yourself? The second and third sentence later after class, you'll copy it again. Okay, the same thing, guys. It's the same exact thing. Remember to write the process. Don't forget about it. You have to write by osmosis, yeah? Can you all write by yourself? After class, you'll write by yourself, okay? The way of movement between organism and amoeba species are different, even though they are both unicellular organisms. Explain this statement. They ask you to explain your answer, why they are different in movement. Amoeba species uses which part to move? Amoeba species uses, correct, the pseudopodium. What about paramecium species? Paramecium species uses cilia. Very good. Okay, one sentence. You have to say, amoeba species move by extending its pseudopodium, whereas paramecium species move through rhythmic beating of cilia. What is rhythmic beating? Yeah. What does rhythmic beating means? Rhythmic beating means you follow up, down, up, down. It's like heart, right? We are also rhythmic beating. Okay, guys, I think I spelled the word wrongly. What is it? R or missing H. Rhythmic beating. Thanks, guys. Okay, no wonder the spelling is so weird. R-H-Y, rhythmic beating. What does rhythmic beating means? Again, it's like up, down, contract, relax, contract, relax. It's called rhythmic beating of cilia. So there's a rhythm. It's called rhythmic beating. You cannot just up. So the cilia have to move up, down. It's called rhythmic beating of cilia. Okay, explain why paramecium species does not require organ systems such as respiratory system found in multicellular. No matter whether they are paramecium or amoeba species, the answers will be the same. Because they uses their plasma membrane, the entire cell can carry out gases exchange. Entire cell. What does entire mean? It means this entire. Entirely. Gases go in, gases go out, gases go in, gases go out. Food goes in, water goes in, water goes out. The entire diagram, the entire cell can carry out gases exchange or maybe nutrients or water absorption. They have a larger total surface area to volume ratio. It means every part is large. Human, we are small because our skin cannot carry out gases exchange. Our skin is just a protective layer. We use our nostril and our alveolus. So our total surface area to volume ratio is very small. Okay, in your answer, we have to say paramecium species is an unicellular organism. with large total surface area to volume ratio. Okay. Next, we're gonna say exchange of gases occur through plasma membrane by simple diffusion. Or you can say it occurs through any part of the body by simple diffusion also can. As long as don't need to have any transport organ. Unlike human, we need to have blood, capillary, we need to have alveolus. They don't have to. So if we are the same size, they will definitely win in the race. Because they can run faster. They can get oxygen faster. Human cannot. Human, we only use nose. So of course, we're going to lose if we are same size lah. Okay, so as I mentioned, the cell organization, I didn't get to do it in this class. So you can go and watch my YouTube video. I teach the last part, or you can just try the question at the back. The answer we posted in the website. Just go to the website, you get the PDF there, you get the answer over there. Gases with GASES and GASEOUS is UK, US, so it doesn't matter. Okay, any question you want to ask, you can ask me now. No, then I will see you guys tomorrow, or maybe I will see you guys when I see you guys again. Okay, so bye-bye, guys. Bye-bye. See you guys. Bye-bye.